So I've been with Genentech for almost 2 decades now, and I'm really excited to be here because I get to talk about 3 things that I really love, and that's manufacturing, innovation, and biopharmaceuticals. So I thought I'd start out with something a little bit fun. I love data, and I'll look for data anywhere, and so I got the nice glossy brochure from IBC when I was putting this talk together, and I went through, and I actually just kind of went through and looked at all the talks and put them in quick categories. So these are my categories to kind of see what we're going to be talking about this week. And I thought this was really interesting to kind of, I've categorized this into antibody design and expression, traditional antibody development. And these are the kind of things I think we probably would have seen a lot more talk about maybe a few years ago. And then some of the really big slices of the pie here are things like viral and microbial controls, manufacturing lifecycle management, where I put all the things like supply chain management, managing CMOs, single-use technology, bispecifics, and antibody drug conjugates. So this is a little bit of a slice of what we've been talking about, and I'm going to come back to this week, and I'm going to come back to this slide in a minute. So mAb industrialization. So this is a really interesting term, which has excited some interesting passion in the industry over the last few years. And so I have a little story to tell about mAb industrialization. So the story's a few years ago, and 2 very interesting things were happening in my career. The first was that after many years of manufacturing, I was transitioning back into the process development realm. The second is that this was the time when, certainly you mentioned my colleague Brian Kelly, as well as the industry was talking about this concept of mAb industrialization and really the maturing of the antibody industry and the creation of platforms, as an example. So these 2 things were happening at the same time. And I remember going to this ACS meeting, and I hadn't been for a few years. I always loved ACS because of the science and technology. Everybody presented there. And I was really excited to go. And I walked out of the meeting feeling just sad. There was kind of a sense of malaise I felt that hung over the meeting because of this concept of mAb industrialization, I think, really left people with a little bit wondering, was there any opportunity left for innovation in the industry? And it was very strange for me having just come from manufacturing, because when you come from manufacturing, this is like the best thing ever, right? Being able to have platforms, manufacturing is all about trying to, bring things in, being able to run things on the same equipment, having standard procedures. And so I spent many years working on a very variable products, many of which weren't even antibodies, and every time the tech transfer efforts were enormous, and the long-term manufacturing of that could be really challenging. And so this fact that we were going to start getting products in that were platform and routine was really fabulous from a manufacturing perspective. But I realized that people that had spent so many years, as an industry, we did take this industry from an industry that had couldn't even make an antibody or didn't know how to make an antibody to one, where now it's a routine, it's a mature industrial industry. And I think we should all be really proud of that, but I can understand how people were feeling a little sad. So the point of my talk today is to get everybody really excited about mAb industrialization. So if everybody walks out here today and this is like your favorite term ever, I'll have accomplished something, because I want to convince you that this really has set the stage to allow us to do some amazing things from a perspective of innovation and for patients. Okay, so the basic, I'm going to go over 4 things. I think the 4 opportunities for innovation I'm going to discuss today are improving our manufacturing consistency, novel drug formats, manufacturing plant design, particularly single use, and drug delivery. Okay, so if I kind of can reframe this data, this is the great thing about non-scientific data, right? You can kind of make it say whatever you want it to say. So I reframed this data along those bullet points. I didn't include drug delivery. This isn't a drug delivery conference, so there wasn't a lot of content there. But you can see a lot of the conversations we are having today, whereas my hypothesis would be maybe 5, 10 years ago, that blue quadrant there, the traditional development, would have been a much bigger slice of the conversation here. It's still important. There's still good work going on there, but it's just a smaller slice of the pie because we are a mature industry. Now we need to really think about how we can manufacture all of these commercial products we have consistently over their lifetime and deliver quality supply to our patients. Thinking about manufacturing plant design, either dealing with our existing infrastructure or looking at innovative new ways to design our plants, and then how are we going to deliver the next generation of drugs in novel formats. Okay, manufacturing consistency. So I actually had a slide for this, and I'm going to skip over the slide, and I'm going to do something that you will almost never see somebody from Genentech do. I'm going to acknowledge that my colleague from Amgen actually did a much better job of presenting this earlier today, and so when I saw him do it, so Cenk Undi actually had a great presentation. Hopefully some of you were there, and he showed some great examples of variability in manufacturing processes, particularly from the lens of raw materials. And so I think he did a very nice job of describing the problem of manufacturing consistency. I think he used the term firefighting at least 3 times in his talk, and that's something. So Amgen clearly has variability in their manufacturing processes. Genentech, we clearly have variability in our manufacturing processes. Having worked in a manufacturing science group for many years, a lot of what you do is firefighting and troubleshooting. And so there is still a major opportunity here. So I'm going to skip over my lame slide. Ask Cenk. He's in the other session right now, so he's not here. But so since this is a problem, if you will all be convinced that this is a problem, what are some of the solutions? And I've broken this into kind of 2 areas. One, which I would call the more traditional process development approaches, and I think some of those were mentioned earlier. Clone selection, I mean, to me, this is a really important one. Once you get your clone, you live with it for many, many, many years. So I think the better you have a clone going in, everything becomes easier downstream. Media development, things like chemically defined media that we have now, fewer raw material investigations, hopefully, from our more complex animal-derived medias. Bioreactor controls, still a lot of great work going on, improving continually the robustness of our chromatography steps. But then there's also what I would call, say, the more manufacturing technology approaches. And these are the things that maybe haven't always had the sexiness of the process development approaches, but when you work in a manufacturing site, these are things that are really critical to your everyday life. And that is things like equipment and facility design, automation, sensors, data technology. I'm going to talk quite a bit about data technology. I've pre-answered Howard's question in my slides, so so I thought that was a great question, so I'll talk a little bit about that. And of course, raw materials and variability. And there's been a lot of great discussions on all of these at IBC this week. Okay, so I was trying to think of how to communicate this. So I went out just to my group. I didn't go and try to engage the whole global Roche network for examples, and said, just give me some quick things that we've worked on to give people a sense that there is innovation going on, and particularly trying to really set up our manufacturing networks for long-term success. So all of these examples are technology examples. I'm not going to talk about people or processes. Those are equally, if not more important. But these are all kind of some quick vignettes on technology. And certainly, if you have questions about any of these, ask me later. Okay, see if I can do it. 10 and under 10. Okay, so the first one. So this was actually very much to Howard's question, global process data management. And so now that we have these very large, complex networks, managing the data is really key. And so this is a screenshot of something that's still in prototype. I don't want to give the impression that Genentech has solved this problem. I wish we had. It's a very difficult problem to solve. But the data technology has just improved tremendously over the last few years. And so the tools available are just like nothing we've ever had before. And so this screenshot, we show some key manufacturing data. And this is for one product. This product's made at 3 different manufacturing sites, 3 different continents. Not surprisingly, 3 different local languages. But we've been able to bring this all into one data system that has a web interface, right? So the data can be accessed anywhere without client tools required. This is a massive, opportunity, and now the technology is actually really feasible because of what's happened with the computer industry. So similar to the global data system, so the global data systems, a lot of that's kind of about monitoring the health of your manufacturing processes and every site is manufactured across the globe. Another way that we're looking at is kind of the longitudinal data, right? And so you have data with your process for years, starting all the way through early development, commercialization, launch, and then hopefully many, many years and decades of commercial manufacture. And so most commercial manufacturing sites now have more modern MES systems that capture a lot of this data, and so we're looking at bringing those even further back into development. So you really have this kind of longitudinal electronic capture of data, once again, to just improve understanding of the products. So those are kind of about getting data. This is about, once you have data, the great thing is you can do some complex analytics. Once again, there's a lot of tools available. If you can imagine what Google is doing with your data right now as we speak, it's really complex, right? And they're doing it, of course, to sell you things, but we can use similar technology to monitor and understand and hopefully predict the health of our processes. And this is one example of multivariate data analysis in this case, it's in a chromatography in our pilot plant. And this is seeing implementations, spotty implementations, but still implementations out in commercial manufacturing. Web-based HTTP, once again, so I think this is a newer technology, not the HTTP part of it. So we've been doing HTTPs for a long time. The first HTTPs I did were, you'd get the little pulse on the column. You'd have a chart. You'd hook up a chart recorder, and then you would get the little chart recorder, and the peak was about this big, and you draw a line. So it's a little older school. Now it's all automated. You have electronic data, to the point now where actually our group has actually put it on a web app. So you don't even have to like turn on your laptop, which is kind of an old school thing these days, right? You can just get it on your web, look at your HTTP, all the calculations are done for you. And so I think there's just a lot of opportunity in these technologies that we will continue to see adoption over the next few years. So, once you have another way, you really want to get good data in, right? So there's analyzing data, but you want to have good data to analyze in the first place. And so there's a lot of been talk, of course, about things like PAT and sensors. I'm not going to talk much about that. I'm really going to talk more about at-line instrumentation. So I think at-line instruments are a really good opportunity because they're much easier to implement generally than sensors, and I think they can give you a much broader array of data to improve your knowledge base. And then once you have that knowledge base, then you can go in and look for a sensor application or a different control mechanism. But really, the first step is understanding your processes. So full disclosure, this is actually a Roche Diagnostics product, this cobas. So I'm not trying to sell it. I don't know if we even get a particularly good price on them, but the reason I'm actually picturing this, because this is something we've recently started doing for monitoring bacterial cultures, and some of the things, and it has a lot of the attributes of what you want when you're looking at instrumentation if you're going to run in an operations environment. It's a robust instrument. These are deployed in the thousands in hospitals all over the place, and so they're run by users that are not necessarily experts. They run in real-world environments. They get beat up. They have technicians all over the place, local technical support to help troubleshoot these instruments. And so this is the kind of instrument that if we really want to move things into real-world manufacturing environments, that these are the types of things that we look for. And of course, that the data meets our needs in terms of what we're measuring. Another example, so it's not just cell culture, purification, there's still opportunities. And so once again, this is not a vendor endorsement, but I think one thing we spend a lot of time in purification is trying to understand protein concentration. It seems pretty simple. And there's two things. One is when you do at-line concentration, you tend to need to dilute it, right? There's a very short linear range, the spectroscopic measurements for doing concentration. And so getting a larger being able to get a larger range without diluting makes it much more manufacturing friendly because you don't have to do the dilutions. But the really exciting part, so this is an at-line instrument. The really exciting potential in my mind for this would be, say, as an on-line instrument, we could finally see the tops of peaks. So for those of you who do chromatography, usually what happens in a manufacturing environment is you cut off the tops of your peaks so you get really good sensitivity right when you start and stop pooling so that you can get good cuts, particularly if you're looking at removing things like product-related impurities. So this gives us the opportunity to both get that sensitivity where we need it, but then actually to really be able to see the full range of your peak, which could help potentially for cutting, but also for troubleshooting would be really great. So even in a very old, well-established measurement, I think there's a lot of opportunity to innovate still. At-line endotoxin is another one. This is kind of the classic QC on the floor. We used to have to submit this to the lab. I will use this as an opportunity to plug poster D5. So poster D5 has this and several other examples from the purification and drug product pilot plant of how we're kind of trying to use these technologies. It improves our workflow in the pilot plant, but also this is the kind of thing that can really relieve burdens on QC labs, and you get data right away, right? You don't have to wait for the data to come back, and so this is definitely a trend.